The instant that the nutrient hit the soil that had been pumped down of water and held at a low temperature for 3 days, we see the curve arising, showing radioactive gas coming out to the tune of about 10,000 counts per minute. Now, we all knew that there's a probability that what we find on Mars is some chemical that will react with the nutrient and produce a curve like that. So the whole biology team debated for years how to differentiate between chemistry and biology. What they came up with was, well, what you do is you heat the sample or a portion of the same sample to a temperature that we think would kill any self-respecting Mars microorganism, that would certainly kill a terrestrial microorganism. You let it cool, you run the experiment again. That temperature is probably too low to damage chemicals that would produce this kind of activity, but ought to show a pronounced effect in killing or damaging the microorganisms. So we did that at California. After we got the test sequence, the active sequence, we heated the control to 160 degrees centigrade for 3 hours, what was selected by the biology team as standard for all the biology experiments as controls. Let it cool, shot the thing again, and you see what we got, zilch, nothing. So that confirmed to all of us that indeed we had a biological sample. And this is what we found in the samples we ran from all around the world. We had never had a false positive in all of the years of preliminary work with this experiment. It went to Mars with a very good pedigree. And what happened on Mars? May we see the next slide. I think the South Pole is down. What happened was very similar to what you saw. As soon as we obtained a sample and injected cycle 1, we had an active curve very reminiscent of the curve that we observed in California in the flight type instrument. So we immediately said, okay, let's go for cycle 2. Let's heat to 160 degrees and run the control. We did, and we got essentially zilch, an excellent demonstration which any place on Earth would absolutely be accepted as positive proof of living microorganisms. We then went to another location, took another sample, cycle 3, and you see the active. At this site, I'm going out of the chronology, but let me explain, we then went into winter, and we could not readily get another sample because we didn't want to repeat the control. We didn't want to waste another chamber. What we did instead was attempt to get another sample. But because of winter's onset, we were told the engineers did not want to risk the sampling mechanism. So we took some of the same sample that gave the active 3 response that had been sitting in the box at 10 degrees C for over 2 months, and we decided we better go ahead and run this thing. Who knows how long the spacecraft will last? We might not make it to spring. And we ran it, and were startled to find no response. So whatever was in that box that we had picked up from the surface of Mars could not withstand sitting around at 10 degrees for a couple of months. That sort of makes a chemical explanation a lot more difficult. 4,000 miles away, we ran a set of experiments on landing site 2 and got essentially the same results. Cycle 1 was the active, and there you see a very strong 15,000 count per minute response. Again, we did not want to go for the 160 degrees C. We're still arguing, can we get a better discrimination between possible biology and chemistry? So we decided, let's have a conference. We got the chemists and the biologists together at JPL, and we said, what temperature would you guys say would discriminate between chemistry and biology in this experiment? And we came up with 50 degrees C. We asked the engineers if they could give us a 50°C heating, and they said they could, and they did. And this was what we got at cycle 2. A very strange response, indicating that there's something wrong. So we asked the engineers to check the instrument. They did. They assured us there was nothing wrong with the instrument. But that is a peculiar response. And what we did following that was to move a sample, to move a rock and get a sample from under the rock, because we began to hear, in addition to chemical reasons that may have produced the response we got, there could be physical reasons, such as ultraviolet light hitting the surface of Mars and creating disjunctions or physical defects in the surface material that would react with our nutrient and produce a false positive. So we went out just before dawn, moved the rock, scooped up a sample, put it into the instrument, and there you see under the rock we got a perfectly good response, somewhat less than cycle 1, but still a strong positive response, which again on Earth would be conclusive evidence. Now we decided, let's try again the 50-degree control. And we did. And we shot it through, and we did get a response. This time it was a 70% diminution of the stronger active. But when we examined the data, we found this curve had this sample had only been elevated to 46 degrees. So you could make a theory that perhaps the organisms were damaged but not killed at 50 degrees, and at 46 degrees they managed to continue metabolizing. But that really is speculation, and I'm more of an empiricist than a speculator, so I'd like to not stress that point.